Hello, students. I'll be with you in these videos just to take you through the mutation lecture. We were supposed to have this lecture in class, but due to the holiday, now we are asked to give it online. So a few things to clarify before I start the lecture. First, as I said, I might split the lecture into 2 or 3 videos. I think that'll be easier for you to absorb. And second, there will be some extra slides on my PowerPoint with some extra information. Those info will not be required from you. It's there only for the sake of clarification. Okay, so let's go through the learning objective of the entire lecture. The main thing we want to try to explain the relationship between change in nucleotide and amino acid sequences. Then types of mutational changes, and then the effect of those different type of mutation on the protein. Would they lead to disease or not? The most important thing for us. And then how those mutations occur. Some of them are spontaneous, some of them intentionally induced or unintentionally. So we need to describe those. And then we just want to know, would all these mutations will be passed to next generation? We need to check that as well. And then absolutely, we are exposing to so many mutagens. Would they be repaired? Do we have a mechanism to repair that? So we need to describe this process and the role of DNA repair mechanism. And then the relationship between DNA damage and cancer, why most of the mutation would lead to cancer eventually. And then we will try to apply what we have learned in the molecular diagnostic lecture to know what are the techniques that we can use to detect this mutation and then analyze the data. Okay. So in this video, I'll try to cover the mutation, its types, and then their consequences or their effect on the protein. So of course, we know by now any change in the DNA-based sequence in any organism is called mutant. But 2 important things that we need to consider. If we consider that change as a mutation, it should be permanent and it should be heritable. And then this mutation that result from damage to the nucleotide of DNA molecule due to mutagen, or sometimes an error, unrepaired error during the replication, can then, within that specific gene, be transcribed into mRNA. Then that mRNA sometimes result in the translation of protein with abnormal amino acid sequence. If you can see the word sometimes there, because there are some types of mutation or nucleotide change that would not have any effect on the protein. And this is an example is silent mutation that we will cover later on. Of course, whenever we talk about the mutation, we just get devastated because we know that it's problematic. We know that most of the diseases are genetic based, and if not, then that mutation might lead to cell death. But at the same time, we need to understand that not all the mutations are bad mutations. Some of them are beneficial, and thus they are the source of genetic variation, evolution, and our existence until today, from millions of years ago. One of the beneficial mutation is when any patient are diagnosed with sickle cell anemia. We know that the mutation itself is terrible, but those patients usually cannot be infected by malaria because that shape of the RBC is not suitable or convenient or does not support the malaria replication. So, as I said, some of the mutations are good, some of them are bad. As we said, mutation is a source of genetic variation. So that means we have some terms like mutant and wild type, whether we are talking about phenotype or whether we are talking about the allele itself. Why we call some certain phenotype wild type? Because it's the most popular or the most common one in the population. For instance, if you look at the blue eyes or brown eyes, is the wild type, because majority of the human being on Earth have a green eye, brown eyes. But the mutant phenotype is blue eyes because a very small percentage of population has an eye color. Likewise for the mutant allele and wild type. The wild type allele is the one that is most popular or most common in population. And then they are not, if we're related to disease, if we're talking about genetic in medicine, they are not causing any problem. We know that we have genes from paternal and maternal side. So we have 2 alleles for a gene. It might happen that both of the alleles that we get from both parents are exactly the same. So we call it homozygous, or it might be different, as we can see in this example. This is heterozygous, or it might be homozygous in case of XY chromosome. The reason I'm trying to explain the homozygous, heterozygous, because there might be some case study that we need to know exactly what does these terms mean to analyze the case. Okay. So whenever we talk about mutation, it can be categorized to small mutation and large mutation. Small mutation, change in nucleotide sequence of single gene. So in the small mutation, we are talking about mutation at the level of gene, not chromosome, not entire genome. While in the large-scale mutation, we are talking about these changes at the level of chromosome. It will be either abnormal number of chromosome like trisomy, or chromosomal structure that are strictly abnormal. It affect the structure. But anyway, the large-scale mutation will not be covered in this lecture. It will be covered by Dr. Sherko in next lecture. But in this lecture, we'll try to cover the small mutation. So type of small mutation. First of all, we want to know, is it only substitution or insertion or deletion? So by substitution or point mutation, we mean that only a single base nucleotide change. And again, the classification or the types of the point mutation itself depend on whether the mutation occurs in the coding region of the gene or the non-coding region. If it occurs in the coding region of the gene, then it could be one of these mutations. It could be silent mutation. By silent, we means that there will be a change in the nucleotide, as we can see here. However, that change luckily would not generate a new amino acid code, rather is the same. So if we look at the coding amino acids code, as we can see, luckily, most of the amino acids have more than one code, except the methionine, because it's the start amino acids. So most of them have different codes. And that's also one of the, I would say, not mechanism, but one of the things that protect us for many years, millions of years, because even if there is mutation, there's a chance that this mutation would not have any consequences because it will still generate the same amino acid. So this is why we call it silent, because there will be no change in the amino acid. We have another type of mutation that's called missense mutation, and this is the mutation that replaces one amino acid with another. As we can see, one point substituted or altered to another, G to C, but this generate, it will replace the amino acid from arginine to proline. Here in the missense mutation, if the new amino acid structurally and chemical property wise similar to the initial one, then we call it a conservative mutation. But if the change is really different, big, in terms of chemical property. For instance, if one is polar and one is non-polar, then this will call non-conservative. And then we also have another type of nonsense mutation that is still in the coding region, and that's called mutation. Again, point mutation that changed the amino acid specified to stop codon. And that's very problematic because, for instance, we have arginine, and after arginine, we are supposed to have so many other amino acids, but because of that change, it might generate one of these three stop codons, and then the whole protein will be stopped. Okay. So again, base substitution can be either transition or transversion. If it's purine to purine or pyrimidine to pyrimidine, that's transition, but the other way is transversion. So as we said, all these different types of point mutation, whether silent, missense, nonsense, is within the coding region. That means within the coding region itself is within the exon, not even intron. That's because they are related to or they are classified, the type are classified based on the new amino acid. Our sequence that is coded for amino acid is only in the coding region. Okay. So this is another explanation for silent, missense, nonsense mutation. In the silent, as we can see, is the same amino acid, and the amino acid followed by this mutation is still the same. For the missense mutation, it might specify a new amino acid. For instance, here we have glutamate, aspartate, but the rest of the amino acid is the same. However, for the nonsense, which is the worst type of mutation, as we can see here, the first is the same, but whatever comes after, or it generates the stop codon, and the protein will be much shorter because none of these amino acids will be generated. Okay, so now we covered the coding if the mutation is in the coding region. But what if we have a point mutation in the non-coding region? Because we know when we talk about a gene unit is not only the coding region. Rather, we have other region that called regulatory and other non-coding region after the stop codon, right? So even here, introns are considered non-coding region within the exons. So what if a mutation happened here or in the capsid or in the regulatory region, whether it's in the theta box, in the promoter, in the enhancer? So for such kinds of mutation outside of the gene can of course also be detrimental, as they can change protein binding site, promoter sequence, splice site, and so on. This is also again for clarification. If some example of mutation in beta thalassemia, there's over 300 different types of mutation that result in beta thalassemia. But as we can see, the region of the mutation are different. Some of them is in the codon region, some of them in promoter region, or the capsid, or the splice junction, and so on. And again, the consequences or the phenotype mutation would be different. By beta 0 mean the protein is non-functional. Beta plus means it's partially functional. And B itself means, which is not mutation, it's totally functional. So as we can see, if it's in the coding region, the point mutation, this is point mutation, it's mostly lead to beta 0. And it depends how important these regions in terms of the protein binding site and active site. Frame shift always beta 0 in the coding region, but if it's in the promoter region, it could be beta plus. That means the protein might still generate, but the quantity might decrease. Okay, so that's why, as I said, even in the coding region, still there might be some consequences, maybe not in the protein structure, or the quality, but rather in the quantity. Okay, so now we are done with point mutation. So we have another type of mutation that called indel, or insertion deletion. This type of mutation is not substitution, but rather we might lose one or even more nucleotide that will be deleted, or one or more nucleotide will be inserted to the DNA sequence. So the other one was substitution, no deletion or insertion, but this one, no. Again, we need to consider, is that indel happen in the coding region or non-coding region? If in the coding region of the gene, we have two types, one of them called non-frame shift, the other one called frame shift. This is correct for both insertion and deletion. So what is the difference between them? Whenever we talk about non-frame shift, that means insertion or deletion of 3 nucleotide or multiple of 3. Why we call it non-frame shift? Because the reading frame of the whole gene wouldn't change. There will be changes in the amino acid sequence, but no change in the frame. Let's have a look. So if we look at this example, okay, and then if we have an insertion of 3 nucleotide, GGT, let's see, 3 nucleotide has been inserted, but the amino acids following that insertion is still the same. Alanine still alanine, glycine is still glycine. Therefore, this kind of non-frame shift insertion or deletion or indel might not have a lot of consequences because only one amino acid inserted. However, in the frame shift, that means indel or insertion deletion of any other number except 3 or multiple of 3. For instance, insertion or deletion of 1 nucleotide, 2, 4, 5. This will result in changing in the reading frame. So beyond the point of mutation, as we can see, the whole things will change. Because we know the codes for the amino acid is 3. So that's why if it's the 3 or multiple of 3, there will be no issue. But if it is less than that or more than that, then everything will shift. So here we have GAA, for instance, glutamine. If the G deleted minus G, minus 1 base pair, as we can see here, ATG is still the same. But after that, it will be AAG and then CAC. As we can see, after the point of mutation or deletion, all the other amino acid has changed. So we will get a protein that is absolutely different from what we are expecting. That's why it might be not functional. Okay. Also, if the indel is in the non-coding region, it might have also effects, and we will talk about this in a minute. But before we do that, let's go through some example or some questions to check the level of your understanding. So I want you to pause the video, go through all the 3 questions, try to answer what you find it the right answer. If you don't know, then go back through the slides. Try to read, to understand, to Google, and then come back to question again. Okay, so I'm assuming right now you did that. Okay, so let's start with the questions. Question 1, it says, type of mutation that affect every codon beyond the point of mutation is... It makes sense to be frame shift. We just talked about that. A non-conservative mutation is the one in which, I would say it's A, the new amino acid has different biochemical property than the original. That's why it's non-conservative. Which type of mutation result in sickle cell disease phenotype? I'm telling you, it's glutamate to valine, so you need to consider their properties. Is it conservative mutation, frame shift? I would say it's non-conservative missense mutation. It's not nonsense because no stop codon has been generated. Glutamate changed to valine, but it's non-conservative because there are absolutely, there's a lot of differences between them, as we can see here. Glutamate and valine change, even if we don't know the property of the amino acid, because of the significant change in the shape of sickle cell anemia, absolutely. The chemical property of these two amino acids is totally different. Okay. Again, for instance, if we have a missense mutation where cysteine changed to tyrosine, because they are both polar, so they function properly. Slightly different, but not, it's like non-functional. But if we have non-conservative where cysteine changed to tryptophan, polar to non-polar, then it can't function properly. There will be some issue. This is what we mean by conservative missense mutation and non-conservative. It could not conserve. Okay. So in a few minutes, I'll try to explain the effects of mutation. We already covered some, but we'll just review in a table and then we'll take a break. I think we also have a couple of questions, so better to take this question as well. Anyways, so coming back to how we assess and explain or predict the effect of mutation, which is the most important thing for us in the medical field. Is it leading to disease or not? So whenever we want to do this analysis, two things to consider. First, types of mutation. Is it point mutation or indel? And the location. Is it in the coding region or non-coding region? So if you came across any type of this question in the exam, just basically consider these. And then if it's in the coding region and point mutation, what kind of point? Missense, nonsense, silent. If it's indel, is it 3 or multiple of 3? In regulatory region, point mutation, which region of the regulatory gene? And then the indel, which region? More importantly, is it small? Importantly, is it smaller indel or large indel? Okay. So let's look at this table: type of mutation in coding region and effect on protein. Silent, because new codon specify the same amino acid, so has no effect on the protein. Missense, possible decrease in function. It's either conservative or non-conservative. We said sickle cell anemia as an example of non-conservative, but again, it decreased in the function, variable effects based on the, as we said, the chemical property of the new amino acid. And we also have nonsense, new codon is stop codon. This is non-functional. This one decreased in function, but in the nonsense is non-functional. Shorter than normal, like factor X deficiency. This is one of the genetic disease. Okay, if indel, we are still talking about in the coding region. If it's non-frame shift, it decrease in function. Again, variable defect effect depends on the function of amino acid added or deleted. For instance, if we have in cystic fibrosis, deletion of 3 nucleotide, which is still 3, it wouldn't change the coding frame, but because that's CTT, eventually would not, that new amino acid would not let the transmembrane protein to bind to the cell surface. That's why it's so problematic. So it decreased in the function. We also have frame shift, endel, again in the coding region. It's usually then functional because the protein will be much shorter than was expected. An example of frame shift in the coding region is Gaucher disease. An inserted single DNA base is only one single DNA base that prevents production of an enzyme. So these were silent, missense, nonsense, frame shift, and their effect. As we can see, missense and frame shift are the worst nonsense and frame shift, they will produce non-functional protein. That's why the clinical sign and symptom will be severe. However, in the case of missense and non-frame shift, this protein will be functional but decrease in the function. Again, mutation in the regulatory region or the non-coding region. Point mutation does not affect quality of protein, rather affect quantity because it's in the regulatory region. So basically, instead of having 100% of the protein, we might get 25%, we might get 70%. It depends when the mutation is. It in the promoter? Is it in the TATA? Because, for instance, if it's in the promoter or TATA box, probably the proteins that needs to bind to the TATA box to start the process of gene expression might not be able to bind properly. That's why we might not get 100% of the quantity that we want. Becker muscular dystrophy is protein dystrophin is normal, but its level are reduced, but still it led to the disease. And again, if it's indel in the non-coding region, does not affect quality of the protein, but again, quantity, like in some cases of beta plus thalassemia, as we saw here. So here in the cup site, there's one insertion, it led to the beta plus, which is, functional protein, but reducing quantity. Okay. So now we knew the effects of different types of mutation. Again, that effect on protein, whether it leads to disease or not. And this is what is important for us. Let's go through this question. This is an example of a question that's the sub-questions, you might have it in the exam, but I will never give you a question in exam as difficult as that. But if you can answer these questions, then probably you absorb the whole material so far. So again, please pause the video, read the case, read the questions, and then write down the answer for you. If you could not find an answer, go back to the previous slides and so on. So I'm assuming that you paused the video, so let's start. Someone has been having recurrent infection and buildup of thick, sticky mucus in his lung. Therefore, the doctor suspected cystic fibrosis. Since cystic fibrosis is a genetic disease, he took a sample and sent it to a molecular diagnostic lab. But he said, I want PCR genetic analysis for the CTT deletion in CFTR gene. CTT deletion, again, over, I believe, 300 mutation in CFTR gene would lead to cystic fibrosis. But the most common one, almost 70% of the mutation, is this CTT deletion. The gene itself is, as I said, encoded for transmembrane protein that maintain the balance of salt and water in and out of the cell, including lung cells. The question gave us the healthy nucleotide sequence, and for cystic fibrosis, and then the protein. As we can see, this is the healthy. And in the cystic fibrosis, the GTT deleted, as we can see. However, the rest is still the same. So let's go through the questions. Although more than 1,000 mutations are identified as cystic fibrosis gene, why the doctor asked for CTT deletion? Why not other 300 or other 1,000 mutations? Sorry, it wasn't 300 for thalassemia. For this one, over 1,000 mutation. So why the doctor ask for CTT deletion? Well, the answer is because it's the most common. 70% of cystic fibrosis patients has that deletion. And because, again, the genetic test is very expensive so far, so the doctor absolutely would go for the very first common one. If then this has been excluded, then it will go to other less common mutation. So by logic, the doctor would need to require the CTT deletion test. Is the CTT deletion a frame shift or non-frame shift mutation? Since it's about deletion, so let's see if it's frame shift or not. Because it's 3, so it's non-frame shift. As we can see, the deletion of this change these 2 amino acids, but the rest of the amino acid are still the same. That's why the reading frame has not been shifted for the rest of the amino acids, so it's non-frame shift mutation. Why the loss of CTT phenylalanine in the protein sequence of that clinical significance? Because that CTT basically responsible of the protein to, we say this is a transmembrane protein to just go through the cell membrane. When we miss that, then that link won't be there. And then the protein will be degraded. Was it the right choice to see PCR technique for detection of CTT deletion? Yes, absolutely. PCR can be used. 2 sets of primer will be generated, one for healthy DNA sequence or gene sequence, and the other one for cystic fibrosis, consider the CTT deletion. And then both can be run. Then once they are run, the PCR result, of course, there will be some probe to the PCR to highlight, and then it'll be run on gel electrophoresis. So PCR is an appropriate technique to be used for such kind of mutation. So question E, since cystic fibrosis is, sorry, just 1 minute. Okay, since cystic fibrosis is an autosomal recessive disorder, according to the PCR, the gel electrophoresis, and the PCR result that we have, which of the sample do you expect to be Saman's sample? So we have to consider a couple of things. It says it's autosomal recessive disorder, and because Saman has all the clinical signs and symptoms, that means both of the allele needs to be infected to have to be infected, not a carrier. Otherwise, you'll be a carrier. So Saman is not a carrier, is infected or affected. So here we have the homozygous, which is normal, and the heterozygous, the carrier, and this is homozygous. How do we know that this is not normal? For instance, let's say if it has not been assigned in the technique. We know that here we have 63 base pair, and because, this is -3 nucleotide, CTT deletion. So as we can see, this one is 16. Because it's smaller, it goes faster and stop later. That's why this is the mutant. Homozygous mutant affected. Okay. That's the last question, then we'll take the break. So here we have Sanger sequencing result for Azat, who is tested for beta thalassemia. We want to know if there's a mutation. The region that there's a difference between normal and mutant, that is Azat's sample, has been shown here. And it tells us 2 things. It tells is a homozygous, and it tells us that the mutation is in the non-coding region, and it says promoter. So accordingly, it says what type of mutation is that, and what does it affect? How does this affect the severity of the disease or thalassemia? So based on what we take in molecular diagnostic techniques, we said that we will have we need to write down the sequence. For instance, here we have 1G, 2G, A, and so on. We will go for the normal, and then for the mutant, I'll just do it like this usually, write down the normal, and then this is the mutant. Then we will compare, see what is missing or what is added or what has been changed, because it could be substitution, insertion, or deletion. So here GG is good, and then A is coming, and then TCC. As if we compare here, TCC as same as this one, and GA and so on. So that means we have only one deletion, one base pair deletion, which is A. So it's one base pair deletion. Because it's in the coding, non-coding region, we cannot say it's frame shift, because there will be no code, amino acid code there. Because it sits in the promoter region, so mostly it will be beta plus. Okay. And beta plus, that means it wouldn't be very severe. It's thalassemia intermediate to beta plus, because the patient is homozygous for the mutation. So it's moderate. Okay. There will be a question in the small group teaching, more questions about beta thalassemia, because it's one of the very common genetic disease in Kurdistan and Middle East in general. Plus, it's a very good tool to study the effect of different types of mutation on different region of the gene. Okay, so I'll stop here. And then the next video will be much briefer. We'll talk about the damage mechanism and how it could be repaired. Okay, so see you soon.